Hello, my name is Michelle King, and this presentation is over gene regulation. So this slide covers the structure of the Lac Operon. The Lac Operon is a well-studied genetic regulatory system in bacteria, notably E. coli, that controls the expression of genes involved in the metabolism of lactose, a sugar found in milk. The Lac Operon is a classic example of how gene expression can be controlled in response to environmental conditions, specifically in the presence or absence of lactose. So let's talk about the structure of the Lac Operon. The Lac Operon includes three structural genes, LAC-Z, LAC-Y, and LAC-A. LAC-Z encodes the enzyme beta-galactosidase, which cleaves lactose into glucose and galactose. LAC-Y encodes the lactose permease protein, which facilitates the entry of lactose into the bacterial cell. LAC-A encodes the transacetylase enzyme, which has a less well understood role in the lactose metabolism. We also have the promoter, operator, and regulator gene. The promoter is the DNA sequence where RNA pol binds to initiate transcription of the structural genes, where the operator is a DNA sequence adjacent to the promoter. It acts as a regulatory element where the Lac repressor protein can bind to block transcription. The LAC-I gene encodes the Lac repressor protein, which can bind to the operator and inhibit transcription of the structural genes. LAC-I is expressed, meaning it is continuously produced by a cell. So what makes the Lac Operon turn on? E. coli bacteria can break down lactose, but it's not their favorite fuel. If glucose is around, they would much rather use the glucose that requires fewer steps and less energy to break down than lactose. However, if lactose is the only sugar available, the E. coli will go right ahead and use it as an energy source. To use lactose, the bacteria must express the Lac Operon genes, which encoded key enzymes for lactose uptake and metabolism. To be as efficient as possible, E. coli should express the Lac Operon only when the two conditions are met. Lactose is available and glucose is not available. So on this slide, we will start understanding the regulation of the Lac Operon. The Lac Operon is subject to both positive and negative regulation, depending on the presence of lactose and glucose in the surrounding environment. So, in the absence of lactose, also known as repression, LAC-I, the Lac repressor, binds to the operator, preventing RNA polymerase from binding to the promoter. Transcription of the structural genes LAC-Z, LAC-Y, and LAC-A is blocked, and the enzymes required for lactose metabolism are no longer produced. So continuing from our last slide, we will talk about regulation of the Lac Operon, but this time in the presence of lactose, so induction. When lactose is present in the environment, some of it enters the bacterial cell through the action of lactose permease, which is encoded by LAC-Y. Inside the cell, lactose is converted by allolactose, a molecule that can bind the Lac repressor, LAC-I. Binding of allolactose to LAC-I causes a conformational change in the repressor protein, preventing it from binding to the operator. With the operator no longer blocked, RNA pol can bind to the promoter and initiate transcription of the structural genes. The enzymes beta-galactosidase, encoded by LAC-Z, and lactose permease, encoded by LAC-Y, are produced, allowing the cell to metabolize lactose. So on this slide, we'll talk about the different controls of transcription initiation. Transcription initiation is a crucial step in gene expression where the information stored in DNA is transcribed into RNA. This process is tightly regulated and involves several control mechanisms to ensure that the correct genes are transcribed at the right time and the right quantities. The primary controls of transcription initiation are achieved through the following elements and processes. So first, let's talk about promoters. Promoters contain specific DNA sequences, including the TATA blocks and transcription factor binding sites. Promoters are typically located upstream of the transcription start site, the point where transcription begins, and their sequences determine the strength of transcription initiation. So let's talk about the transcription factors next. Transcription factors are proteins that bind to specific DNA sequences within the promoter region and regulate the initiation of transcription. There are two types of transcription factors involved in initiation. The first we'll talk about will be the general transcription factors, or GTFs. These are essential for the transcription of all genes and include proteins like the TATA binding protein, TBP, and the transcription factor 2 complexes. GTFs help RNA polymerase bind to the promoter region and form the transcription initiation complex. So next, let's talk about specific transcription factors. These are regulatory proteins that bind to the enhancer and silencer sequence, often located far from the promoter. Specific transcription factors can either enhance, activators, or inhibit, repressors, transcription initiation by interacting with RNA polymerase and the GTFs. Lastly, on this slide, let's talk about RNA polymerase. RNA pol is the enzyme responsible for synthesizing RNA from a DNA template during transcription. In eukaryotes, RNA polymerase II is primarily responsible for transcribing protein-coding genes. It plays a central role in transcription initiation by binding the promoter region of the gene. So let's continue with control of transcription initiations. So next we'll talk about the mediator complex. The mediator complex is a large protein complex that acts as a bridge between transcription factors bound to enhancers or silencers in RNA polymerase at the promoter. It helps facilitate communication between distant regulatory elements and the transcription initiation machinery. So let's move on to chromatin structure. The accessibility of DNA in the promoter region is influenced by chromatin structure. DNA is wrapped around histone proteins to form nucleosomes, and tightly packed chromatin can inhibit transcription initiation. Chromatin remodeling complexes and histone-modifying enzymes can alter chromatin structure to make the promoter region more or less accessible for transcription. So now let's touch on epigenetic modifications. Chemical modifications to DNA, such as methylation, and histone proteins, such as acetylation or methylation, and phosphorylation, can also impact transcription initiation. These modifications can serve as marks that either enhance or repress transcription by affecting chromatin structure and the binding of transcription factors. Then let's lastly on this slide talk about signal integration. Many genes are regulated in response to environmental signals or cell signaling pathways. Signaling molecules can activate or inhibit specific transcription factors, altering their ability to bind to DNA and to initiate transcription. Okay, so let's move on to genomic imprinting. Genomic imprinting is an epigenetic phenomenon in genetics that results in the differential expression of genes based on the parent of origin. In other words, certain genes are marked or imprinted with chemical modifications that affect their activity, and these imprints are inherited from one parent and silenced in the offspring. This process leads to monoallelic or single copy expressions of specific genes depending on whether they were inherited from the mother or the father. Genomic imprinting is characterized by parent of origin specific gene expressions. This means that the expression of imprinted genes is determined by whether the allele was inherited from the mother or the father. Let's move to number 2, epigenetic marks. Imprinting involves epigenetic modifications, primarily DNA methylation, that are added to specific regions of genes and during gametogenesis, which is the formation of sperm and egg cells. Number 3, monoallelic expression. Imprinted genes are typically expressed from one or two alleles. The other allele is silenced through epigenetic marks. Now, functional implications. Many imprinted genes play critical roles in development, growth, and behavior. Imprinting can influence traits and diseases in humans and other organisms. So I've included a couple examples of some imprinted genes. So let's talk about our IGF-2. IGF-2 is a peptide hormone and a member of the insulin-like growth factor family. It is produced primarily in the liver and the other tissues. IGF-2 is involved in promoting cell division and growth in various tissues throughout the body. It acts as a mitogen, stimulating cell proliferation. It is particularly important during fetal development, where it plays a crucial role in regulating the growth of various organs and tissues. It is one of the key factors controlling prenatal and postnatal growth. IGF-2 expression is subject to genomic imprinting. In mammals, including humans, only the allele inherited from a specific parent, usually the father, is actively expressed, while the other allele is silenced. This imprinting is regulated by epigenetic mechanisms such as DNA methylation. Now let's talk about H19. The H19 gene is a non-coding RNA gene that plays a significant role in gene regulation and development. H19 is located on chromosome 11 in humans. It codes for long non-coding RNA molecules. Like IGF-2, H19 is subject to genomic imprinting in mammals including humans. H19 is typically expressed from the maternal allele while the paternal allele is silenced. This in contrast to IGF-2, which is expressed from the paternal allele. The regulation of H19 and IGF-2 imprinting is controlled by differentially methylated regions known as the imprinted control region or ICR. Methylation status at this region determines which allele is active and which will be silenced. The specific function of H19 is still an active area of research. It is believed to play a role in regulating cell growth, differentiation, and development, possibly by acting as a precursor for small regulatory RNAs or by influencing the expression of other genes. So lastly on this slide, let's talk about PEG3, known as paternally expressed gene 3. This is a gene that plays a significant role in genetic imprinting and is involved in various biological processes. So, PEG3 is located in humans on chromosome 19. It is classified as paternally imprinted gene, meaning that it typically is expressed only from the paternal allele, while the maternal allele is silenced. Like H19 and IGF-2, PEG3 is subject to genomic imprinting. The specific regulations of PEG3's expression is controlled by epigenetic modifications such as DNA methylation, which ensures that it is expressed from only the paternal allele. The exact function of PEG3 is still an active area of research. It is known to be involved in various processes, including embryonic development, growth regulation, and behavior. In mice, mutations in PEG3 has been shown to lead to defects in maternal nurturing behavior. So now let's move into applications and implications of genomic imprinting. So number one, let's talk about genetic disorders. Disruptions in imprinting can lead to genetic disorders. For example, defects in imprinting at the 15q11 to 13 region are associated with Prader-Wills syndrome, loss of paternal alleles, and Angelman's syndrome, loss of maternal alleles. Number two, development and growth. Many imprinted genes are involved in embryonic development and the regulation of fetal growth. Understanding imprinting can provide insights into development disorders and growth-related diseases. Now let's move on to behavior and neurobiology. So some imprinted genes are associated with behaviors and neurobiological processes. For example, imprinted genes have been linked to social behaviors and brain development. Lastly, let's talk about cancer. Changes in imprinting patterns can be involved in cancer development. Altered imprinting of certain genes can contribute to the uncontrolled growth of cancerous cells. So let's talk about the timing of genomic imprinting. Imprinting occurs during gametogenesis, specifically during the development of sperm and egg cells. This process establishes the epigenetic marks on imprinted genes. These imprints are maintained and passed on to the next generation when the sperm and egg fuse during fertilization. Imprinted genes maintain their parental specific expression patterns throughout an individual's life. So to summarize, a genomic imprinting is an essential epigenetic process that affects gene expression based on paternal origin. It has broad implications in development, growth, behavior, and health, and it is established during gametogenesis and maintained throughout an individual's lifetime. So now let's talk about post-transcriptional control. Post-transcriptional control in genetics refers to the regulation of gene expressions that occurs after the process of transcription has taken place. Transcription is the synthesis of mRNA from DNA, and it is the first step in gene expression. Post-transcriptional control mechanisms allow cells to further fine-tune and regulate gene expressions by influencing the face of mRNA molecules before they are translated into proteins. Here's an explanation. So let's start with mRNA splicing. In eukaryotes, pre-mRNA undergoes splicing where introns, non-coding regions, are removed and exons, the coding regions, are joined together. Alternative splicing can generate multiple mRNA isoforms from a single gene. So let's talk about RNA editing. So some mRNA molecules undergo editing when specific nucleotides are modified after transcription. This can change the coding sequence and affect protein function. So now let's move on to stability. The stability of mRNA molecules can be regulated. Some mRNAs have short half-lives and are rapidly degraded, while others are more stable and persist longer in the cell. And lastly, on this slide, we'll touch on RNA transport. So mRNA molecules must be transported from the nucleus to the cytoplasm for translation. This process is tightly regulated, and certain mRNA molecules may be selectively transported or retained in the nucleus. So let's continue. So mRNA localization. In some cases, specific mRNAs are localized to particular regions within the cell, such as the dendrites of neurons. This localization can impact local protein synthesis and cellular function. Let's talk about some of the modifications. Post-transcriptional modifications, such as the addition of the 5' cap and the poly A tail, are crucial for mRNA stability in translations. These modifications also facilitate ribosome binding. Let's talk about our microRNAs and our small interfering RNAs. The small RNA molecules can bind to the specific mRNA sequences and either inhibit translation or promote mRNA degradation. Post-transcriptional control plays a crucial role in gene regulation, allowing cells to respond dynamically to internal and external cues, fine-tune protein production, and achieve precise control over various cellular processes. Dysregulation of these mechanisms can contribute to various diseases and developmental disorders.